Kia ora everyone, welcome to the next of the series of mini lectures, and what we're going to look at in this one is the benefits that class 1 endophytes or epichloi endophytes provide to their host plant, and in particular the role that secondary metabolites play in providing those benefits. And we're going to start to look a little bit at the evidence that these endophytes provide benefits and that these secondary metabolites are the agents that are responsible for those benefits. So as usual, I'm going to switch off my video and we'll get started going through the slides. All right. So at the end of the last mini lecture, I said that the reason that we consider these endophytes to be mutualist is because they provide benefits to their host plants. And it's thought that the endophytes in particular produce beneficial compounds that provide benefits to their host plants. And so we're going to look at those. And often these compounds that they produce are what we call secondary metabolites. So secondary metabolites are non-essential metabolites, so chemical compounds produced by fungal metabolism that are not directly involved in normal growth, development, or reproduction. And so metabolites or chemicals that are produced by fungi that are involved in growth, development, or reproduction are called primary metabolites. And you can think of these secondary metabolites as being kind of auxiliary features that the fungus produces to give it extra features, I guess. And the epichloi class 1 endophytes produce numerous secondary metabolites that we think are important for them to provide benefit to their host grasses. And the best characterized ones of these are compounds that deter grazing insects. And if you're a farmer, this is a good thing, because if you've got grass that you want your stock to eat, then you would prefer that the grass is not eaten by grazing insects, so it's therefore available to be eaten by your sheep or cows or whatever it is. But they also produce compounds that deter grazing mammals, which of course is not as good if you are a farmer. And other secondary metabolites may protect against fungal pathogens and against conditions such as droughts or floods. And the idea here is that these secondary metabolites are producing these benefits that offset the costs of fungal nutrient consumption, so the use of plant sugars by the fungi, and also potentially of things like the pathogenicity, so the suppression of host grass reproduction that we talked about in the previous lecture. And so by providing these benefits, they're able to offset these costs and therefore act as mutualists for the benefits of the plant. So let's go to the next slide. And in this next slide, I'm showing some examples of some secondary metabolites from the epichloi endophytes. You do not need to know these structures, they're just there for illustration. But if we start in the top left, this is ergovaline. And ergovaline is a compound that protects against grazing mammals. And it's actually related to a toxin that you may have heard of called the ergot toxin that affects humans. And so this was famous in the Middle Ages, where people would harvest rye, where the rye seeds had an endophyte, a class 1 endophyte, not actually an epichloi endophyte, one from a different genus, but a related endophyte, that was growing in those grass seeds. And that endophyte produced a compound called ergot, which is related to ergovaline. And that caused tremendous toxicity in humans, including hallucinations and enormous pain, and eventually dropping off of limbs. And we'll look at a similar effect in grazing mammals in the next slide. Down at the bottom left here, we have something called lolotrim B, and this is an example of a large class of chemical compounds called the lolotrims. And these lolotrims also protect against grazing mammals, and we'll look at an example of that. And then finally on the right here, we have pyramine at the top and an example of a lolene. So again, lolenes are a large class of chemical compounds, and the pyramines and lolenes are thought to protect against grazing insects, and we'll also look at that. So going to the next slide, slide number 3. So I've said that these chemical compounds produce these effects, but what is the evidence? If we want to think like scientists, then we want to understand We want to see the evidence more directly. And so here's some examples. So what's shown here is what's known as fescue foot. Fescue is a kind of a grass, and this is caused by ergovaline toxicity when the animals eat endophytes, that is, or eat grass that has endophyte that's producing ergovaline. And so one of the things that ergovaline does is it causes vasoconstriction, so loss of the blood supply to the limbs, and they will eventually grow gangrenous and fall off. Not particularly pleasant. What I'm going to do is I'm going to try and break out of here and show you a video of sheep that have a condition called ryegrass staggers, and that's what happens if they eat grass that has endophyte that produces lolotrins in them. So I'll see if I can swap screens here. Bear with me, it may take a little while, and we'll have a quick look at this video. Okay, and so the final example that we look at here is the effect on insects. And so here we have some grass. So the one on the left doesn't have endophyte, the one on the right does have endophyte. And this grass has been infested with an insect pest called Argentine stem weevil. And you can see that if you have the endophyte, so the one on the right, there's great protection against grazing by those insects compared to the grass on the left, suggesting that the endophytes are protecting against the insects. Okay, so if we go to the next slide, slide number 4, I've given you anecdotal evidence to support the idea that these endophytes and the secondary metabolites they produce protect the grass against grazing of these various types. But is there actually hard scientific data to support this? That circumstantial evidence is not really as convincing as it should be if we want to think like scientists. And so what I want to do is I want to describe an experiment that people did to look at whether a secondary metabolite is causing protection by the endophyte for its host grass to insect grazing. And they did this experiment using perennial ryegrass, which is a major New Zealand pasture grass, and they fed leaves of this ryegrass to that Argentine stem weevil that I talked about in the previous slide. And this is a major pasture pest in New Zealand. And so what they did is they took ryegrass leaves where in some cases the ryegrass had endophyte present, and in some cases the endophyte was absent. And they measured the level of leaf feeding, which is shown on the graph over here. And then they also took photos of the actual leaves, and you can see the feeding is these white patches appearing here. And so what you can see is that when the endophyte is present, there's much lower levels of leaf feeding compared to when the endophyte is absent. So this is pretty good evidence that and so this is the Epichloe endophyte, that the Epichloe endophyte protects against grazing by Argentine stem weevil. But what this doesn't do is it doesn't provide any evidence that the protection against Argentine stem weevil comes from the production of secondary metabolites by these Epichloe endophytes. So for example, it could just be that the Argentine stem weevils don't like the taste of fungi, and that's why they don't feed on these things. And so what the scientists who did the study then did was they looked to see whether it was the secondary metabolite pyramine that I talked about previously that was preventing or deterring these stem weevils from grazing. And to do that, they took a gene that was responsible for making pyramine and deleted it from the endophytes, and then looked at the effects. So pyramine is made with a biosynthetic pathway composed of a bunch of enzymes, and those enzymes are encoded by genes. And so they deleted one of these genes that then meant the Epichloe endophyte couldn't produce pyramine. And they have 2 mutants like that over here on the right. And what you can see is that when the endophyte is no longer able to produce pyramine, the level of leaf feeding is more or less the same as you see when there's no endophyte at all. And down here you can see that the Argentine stem weevils have grazed on these leaves, and you can even see some of the weevils here. So this is really good evidence that not only is the endophyte protective against grazing by Argentine stem weevils, but it's the production of paramine that's the important thing that stops these weevils from eating the grass. So hopefully that logic is clear. All right, let's go to the next slide. And so here, what we're looking at is the idea that you can select endophytes that are ideally suited for pasture grass conditions. And so people in New Zealand, as well as overseas, are trying to develop endophytes that have the best combination of secondary metabolites for pasture grasses. So as you might imagine, these would be endophytes that produce compounds that deter grazing insects, but don't deter grazing mammals. And they may also have other advantages, such as deterring other pests, such as nematodes, and also being protective against things such as droughts and floods. And this approach involves an understanding of the genetic basis for secondary metabolite production. So as I said, the secondary metabolites are produced by biosynthetic pathways, where the enzymes are encoded by genes. And very fortunately for us, the genes that are involved in, and so there's usually multiple genes involved in making a single secondary metabolite, but these genes are grouped together in the genome in what we call clusters. And so you don't need to know the details of this picture here, but each of these lines represents part of the genome of a different class 1 endophyte, and the little arrows reflect different genes. And the ones that are kind of shaded in blue here, these are one particular set of genes that's involved in producing one particular secondary metabolite. And so by looking in the genome of these fungi, you can see what genes are present, and therefore perhaps which secondary metabolites each fungus is able to produce or not able to produce. And so you can select for a particular endophyte species that has a good combination of secondary metabolites for what your particular application is. Okay, and then I'll just go to the final slide for this mini lecture. And here I want to come back to the idea that there's a continuum between mutualism and pathogenicity. And so whether the benefits provided by a fungus through, say, the production of secondary metabolites outweigh the cost it imposes through the use of sugars and other things such as sterilizing the grass flowers, that determines whether it's a mutualist, a commensal, or a pathogen or a parasite. And so where on the continuum between pathogenicity on one side and mutualism on the other a fungus falls depends on the balance of benefits it provides versus the costs that it imposes. And so we can think about this balance as kind of being like a seesaw, as is shown here. So if the seesaw is perfectly balanced, as is shown in the diagram here, then we can think of this as a commensal relationship. It neither benefits nor disadvantages the host grass. But imagine that the costs start to outweigh the benefits. Now the seesaw will tilt down towards the parasitism part of the spectrum, and so the fungus will be a parasite or a pathogen. Imagine, however, that the benefits outweigh the costs, and so now the seesaw tilts in this direction here, and now we're in the mutualism spectrum, and so we can consider the fungus to be a mutualist. And so by looking at whether the benefits outweigh the cost or vice versa, we can see whether it's a parasite or a mutualist. And the extent to which they outweigh one or another depends on where on the continuum between parasitism and mutualism lies. So if the benefits and costs are very closely balanced, it might be slightly parasitic, slightly mutualistic. But if they're very unbalanced, then it might be a very mutualistic or a very pathogenic fungus. So hopefully those ideas are clear, and we'll come back to that a little bit more as well in subsequent mini lecture. But for the moment, I'll wrap that up. And again, if you have any questions over any of this, please post them on Piazza.